vision research, and today is no exception. So I would like to welcome all of you and also our speaker who traveled long distance and join us in person, and is a wonderful scientist. So we're very happy to have you here today. But formally, let's get Yasmin to introduce you.
Welcome everyone to today's seminar. My name is Yasmin Solano and I'm a PhD candidate in Dr. Philip Kaiser's lab. Today I have the pleasure of introducing our in-person guest speaker, Dr. Machelle Pardue. Dr. Pardue is a multifaceted scientist who currently is a professor and the vice chair of research in the Department of Ophthalmology at Emory University and a senior research career scientist at the Atlanta VA healthcare system. Dr. Pardue received a Bachelor of Science degree in zoology from the University of Wyoming, and she then received her doctorate degree in vision science and biology at the University of Waterloo. Her postdoctorate training was in visual electrophysiology at Loyola School of Medicine and Hines VA Hospital in Chicago. Dr. Pardue has been part of the intramural research program at the Department of Veterans Affairs for her entire career. Dr. Pardue serves on multiple review boards, including national, international, regional, and institutional grant review boards. She serves as a manuscript reviewer for over 23 journals. Dr. Pardue has spearheaded the organization of conferences, including one of our favorite, the ARVO annual meeting. And throughout Dr. Pardue's career, she has published over 130 manuscripts, and this does not include her review and book chapters. She has been named a Gold Fellow for the Association of Research in Vision and Ophthalmology and a Fellow of the American Institute of Medical and Biological Engineers. Dr. Pardue's lab is focused on clinically relevant treatments for retinal disease that can make a difference in the quality of life of patients. Currently, her laboratory is developing the novel screening and treatment strategies for early stage diabetic retinopathy, elucidating the retinal mechanisms of myopia, and developing neuroprotective strategies for retinal diseases. She is moving several preclinical studies to the clinic to create novel treatment strategies for patients. So today she will present a talk titled Using Functional Biomarkers for Early Detection and Treatment of Diabetic Retinopathy. So please help me welcome Dr. Machelle Pardue.
All right, excellent. Well, thank you so much for that introduction. It's a pleasure to be here today. I always love coming in person to different locations and being able to meet people and visit and see the location. So it's really wonderful to be here. And this is the first time that I've been to Irvine. So, so thank you for this invitation. So let me start here with my title slide and tell you what I'm going to speak to you about today. And that is really about diabetic retinopathy. So the scenario where we have patients who are developing diabetes, and then they are getting retinal vascular lesions and vision loss. And what I'm going to tell you today about is a biomarker, a functional biomarker that we've discovered that seems to be able to detect the disease earlier than these vascular changes, these vascular lesions. And when we can detect things early, that also means we have a new treatment window where we can actually try to intervene. And what I'm going to propose is that if we can do early detection and treatment, then we are actually going to be able to prevent these vascular lesions and this vision loss. Let me just put my pointer up here. Let's see. There we go. All right. So I do have a conflict of interest in that I have a patent pending and a company that's licensed the technology. So I'm sure that this audience is well aware that diabetes is a huge problem. And so here in this graph, you can see that diabetes has steadily increased. And this data goes from 2000 here to 2019 with 463 million people that are affected worldwide. But what's scary is that the projections have actually been outpacing the actual reality. So here in the orange, you have some projections for various years, and you can see that the projections have already, we've already outpaced most of these. And so this is a huge problem in the US as well as worldwide. So about 20% of people with diabetes have this complication of diabetic retinopathy. And in the US, that means that we have over 7 million people that have DR. So it is a leading cause of vision loss in the US and across the world. But what is, kind of staggering is that diabetic retinopathy is estimated to more than double by 2050. And that is data from NEI. So how do we currently detect diabetic retinopathy? Well, we do this with actually a very well-established screening program that I'm sure you guys are aware of, where patients who have diabetes are told to go in and get a yearly eye exam, really a fundus exam, so that they can try to detect changes that are happening in the retina. And those changes include hemorrhaging or these cotton wool spots or neovascularization that would indicate that there's these vascular changes going on in the retina. So in the US, there are over 34 million people that have diabetes. That means that many people need to go and get this sort of an exam every year. Now, that's a lot of people, right, that need to have an exam, and a lot of them don't go. So, maybe you know of people like this that are like, that's not going to affect me. Maybe they're young, they have diabetes, and they're still in their 20s, and they're like, yeah, there's no reason I should go, and they don't go. So I actually know of an individual who he was an undergrad, and he had not gone to get his eye exam, went for the first time as an undergrad, and was driving home from that exam and had to pull over to the side of the road as he's talking to his mom because he's crying because he has been diagnosed with diabetic retinopathy, right? So it is a serious condition, and there's not a whole lot of treatments out there that are really going to stop that progression and make a difference in the vision loss. So, all right, so we have a screening program, and a lot of people need to sign up for it. But when we look at, this kind of progression of this disease here on this timeline, you can see that people get hyperglycemic, they get diagnosed with diabetic retinopathy, and these retinal vascular lesions actually take 10 to 15 years to develop. So there's this huge window here where we're just kind of monitoring people, waiting for them to develop these vascular lesions. And so we really wanted to know, well, can we have earlier detection? Like, surely there's something here in this 10 to 15 year window that we could detect that might be some sort of biomarker. And as I said, if we have an early biomarker, we can have treatment, and then you could have prevention. So my lab set out to investigate this, to be able to say, well, what is happening prior to these vascular structural lesions? As you heard, I was trained in electrophysiology during my postdoc, so I really think a lot about functional assessments, functional biomarkers. And so we decided to use the electroretinogram and also look at visual acuity and contrast sensitivity. And the other important aspect about these two choices is that they're both things that we could also measure in the clinic, right? So very much thinking about the translational power of the things that we're going to assess. All right, so even though we think of diabetic retinopathy as a vascular disease, there's more and more evidence that there's actually neuronal changes that are happening. And as you can see in this slide, ERG defects have been reported in a number of different studies. So you can see the ERG A wave and B wave amplitudes or implicit times, the oscillatory potential implicit times or flicker implicit times have all been reported to change. And this is both in diabetic patients as well as in diabetic rodents. So it's not that we don't know that there could be some functional changes that are happening. And some of these were reported in conjunction with vascular changes, and some of these studies were before vascular changes were detected. Now, I'm going to very much focus on the part of the ERG waveform that are these oscillatory potentials. So here you can see the retina, just to kind of give you an idea of the cell layers that we're recording from. And here you have a series of ERGs from a very dim flash to a very bright flash. And this ERG waveform has a very characteristic shape. We have a negative going A wave, a very positive going B wave, and then you have these little wavelets here. And these wavelets are called oscillatory potentials, and they're actually generated by the amacrine cells mainly, but also the late ones here are also, there's evidence that they might be generated from the retinal ganglion cells. Now, the other part of this ERG that I want to emphasize to you is the cell types that are really generating this response. So these waves that I'm showing you are all from a dark adapted retina. So the retina is dark adapted, and then we start with these really dim flashes and we go to brighter and brighter flashes. And when you have a dark adapted retina and you present a dim flash, you're really only stimulating rod pathways. Okay, so those very dim flashes, rod only pathways. And as we get brighter, then we have these mixed rod and coast pathways that are being stimulated. And that's going to be an important thing to keep in mind through this talk. Okay, a lot of the data I'm going to show you today is from rodents. And so how do we do ERGs on a rodent? This is a rat, it's a long Evans rat. You can see that we have them sitting here on a heating pad. We put electrodes on the cornea. It could be a DTL fiber electrode, so a nylon fiber that has silver particles in it, or a gold loop electrode. And then we have needle electrodes for the reference and a needle electrode that's back here at the tail. And we're going to make our animals diabetic using STZ or streptozotocin. So STZ is going to kill the beta cells that are in the pancreas and make the animal hyperglycemic. And it's considered a type 1 model of diabetes. So in the rats, we do this with a single injection of STZ. It's a pretty high dose. It's definitely a toxin, so the animals do get sick and visibly sick, and they lose weight with this. Or with mice, we can use a multiple low dose type of regime where we have 5 consecutive days at a lower dose. We consider hyperglycemic status to be when the blood glucose is over 250 milligrams per deciliter. And we do give insulin to our animals. So if they've lost more than 10% of their body weight, then we do give them a low dose of insulin. But it never changes their glucose levels to change the fact they're hyperglycemic. So they're hyperglycemic throughout the study. Okay, the other thing that we're going to be measuring today, I'm going to show you, is the optomotor response. And we use this in order to measure spatial frequency and contrast sensitivity thresholds. I'm sure many of you are already familiar with this. We put a rodent in the middle of a chamber, which consists of 4 computer monitors, and we basically create a virtual drum that rotates around the animal. And if the animal can see the stripes that are rotating, they will do a head turn in that direction. So this is a reflex. It works really well. I'm always impressed at how reliable it is for measuring spatial frequency and contrast sensitivity thresholds. And we do this test at baseline, and then we do it every other week after hyperglycemia is confirmed. Okay, so let's move on to some data. So we were looking at being able, like a wide range of flashes when we started, to be able to see where we could potentially see any change in a diabetic animal. And we did this starting, just a couple weeks after they had had the STZ, they're hyperglycemic, and we're like trying to track, okay, when do we first see something? So this data I'm showing you here is from diabetic mice. This is 5 weeks after the STZ injection. And I'm showing you examples from a dim flash as well as a bright flash. Now, these are just the oscillatory potentials. The black line is the control, and the red lines are the diabetic. And what I want you to notice first up here is that there's this gray vertical line, and that indicates OP2 in the control. And it goes all the way down through the diabetic, because what I really want you to note is that the diabetic is delayed. And so when there's a delay, I'm going to put a little arrow where the similar OP wavelet is, okay? So it should be very obvious, okay, this is where the control is and this is where the diabetic, and you're gonna see this in multiple figures. So you can see in the dim flash, we definitely have a delay, but in the bright flash, we don't see a delay, okay? And let me show you here in an intensity response function. So I'm plotting amplitude over luminance for each of these OP wavelets, OP1, 2, 3, and 4. And you see that amplitude is affected if we go to OP4, the very end OPs, when we get up into the bright flashes. But look here at the implicit time. So in the implicit time, if we look at the intensity response function, you can see that we have a delay at just the brightest, just the dimmest flash intensities. And that is true for all of these OPs. But notice that once you get up to kind of the mixed response with both rods and cones responding, then there's no delay. So this was the first thing that we saw. And it was like, well, this is interesting, right? So you actually are seeing something that's only in the dim, and you have this very, recognizable delay. So again, we're going back to the literature and saying, okay, well, has this ever been seen before? Well, yes, it has, and that ERG OPs have been reported to be delayed, and particularly in diabetic patients. It was first reported back in the 1960s. And many other studies have reported OP delays, but importantly, they all use bright flash stimuli. And if we look at this figure here, you can see the different stages of diabetic retinopathy, from no retinopathy all the way to proliferative retinopathy, which is kind of the end stage with vitreous hemorrhages. And these are the OPs from a patient and from several patients. And you can see that these are the OP wavelets. And again, I've got my vertical line here, so you can see where there's a delay. So you can see as we go through this different stages of diabetic retinopathy, the response gets more and more delayed. And in fact, when you get down here to the very end stage, you can see that these oscillatory potentials aren't even recognizable, right? So they basically aren't even there. Okay, but all of this is with a bright flash. Okay, so definitely can see ERG changes when you have vascular defects that are also present. Okay, well, we are going back to our animals now, and we're trying to look at this more in progression across diabetes. So we take, now I'm showing you some rats. The top line is the control. And then these are rats that have been followed at 4, 8, and 12 weeks post-STZ. So I need to tell you that rats at these ages do not have vascular defects. And in fact, most of the time when we think about rodents having vascular defects, it's not until 4 or 6 months after they're become hyperglycemic. So quite a while. But there is a caveat here in that they never develop the neovascularization that we see in people, but they do develop leakage and pericyte dropout. So, but at this stage, there's no vascular changes. I'm showing you the dim flash, and you can definitely see that there's a delay in the diabetics, and this delay gets more and more delayed as we progress. Now, if I show you the bright flash, you can see that we don't see a delay until the 12-month time point. Or I'm sorry, the 12-week time point. So it took a lot longer to be able to see this changes in the mixed rod cone response. So our take home from this was that this rod-driven intraretinal response, which is where these OPs are coming from, was affected first in diabetic retinopathy. And this work was really driven by an MD PhD student in my lab at the time, Harry Ong. Now, Harry also looked at the visual acuity and contrast sensitivity from these rats, and you can see that that also was affected very early. And in fact, right away, you can see that in looking at this visual acuity or spatial frequency thresholds, the diabetic animals have a decrease, and it just continues to decrease across these 12 weeks. And we also are seeing a deficit in contrast sensitivity as well. All right, so I really want to make sure that I emphasize to you how this is kind of seems to be progressing between the very dim versus the brighter flashes. So the difference between rod-driven and some sort of a mixed response. So here at 7 weeks, I'm plotting the diabetics here in the red line. And then the dotted black lines are the 95% confidence intervals for our controls. And notice here in the dimmer flashes, you can definitely see that there's a delay. It's not actually not significant right here in this particular cohort. But then when you get up to the brighter flashes, there is no difference. Now at 14 weeks, this is definitely significant, but notice that the significance disappears as we get brighter. At 20 weeks, more and more of the flash intensities are involved, including at 29 weeks, it keeps getting, more and more advanced. So indicating that those rod pathways are definitely being affected, but the rest of the retina, the rod cone pathways, are also being affected as the disease progresses. All right, so what do we know about these rod pathways? They have been reported to have changes in diabetic retinopathy. So we know that they have very high metabolism, and that may actually induce hypoxia in the inner retina, and that might contribute to these vascular defects. Other folks have also reported changes in these rod-driven inner retinal dysfunction, like looking at endocrine cells. And we also know that there's some reports of changes in the GABAergic endocrine cells in diabetes. And so that could also be involved here. And finally, endogenous insulin signaling in the RPE contributes to the rod photoreceptor function. And so that could be another source of why we have rod pathway defects. Now, we had these results, and they were intriguing, but they're all in rodents, right? So we really need to know, well, do we see a similar type of thing in patients? And one of the challenges with the patients is that those oscillatory potentials I've been showing you look great in rodents. They're really large, easy to measure, but in patients, they're a lot harder to see. And so these are just the example ERG waveforms from what is a dim flash and a bright flash. And I just want you to first notice that these oscillatory potentials don't stand out nearly as well as in the rodents. Now, what I'm showing you here is 4 different responses from 4 different flash intensities. And first, we wanted to use what's called the ICEV standard, dim and bright. So ICEV stands for the International Society for Clinical Electrophysiology in Vision. It's a mouthful. So ICEV, and they put out standards of how you should run an ERG that most clinics use. And so a typical human ERG would use an ICEV dim and an ICEV bright to record an ERG. Now, we were concerned that this ICEV dim would be too dim to actually be able to see our oscillatory potentials. So we added it to test flashes. Is there a question?
Yeah.
Okay. So, and then the test flash here, you can see that it's a little bit brighter, and negative 1.4 log candela seconds per meter squared. And then we have another test flash at a negative 0.8. And then the ICEV bright is 0.4. And we're going to look at patients that are diabetic and also ones that have already been diagnosed with diabetic retinopathy through fundus exam. And so here's just some of the statistics on these particular individuals. You can see their HbA1c levels, duration of diabetes, and whether or not they were taking medication. So the different lines here, the black is the controls. The red is this diabetic group that doesn't have retinopathy. And then the blue line is the diabetic retinopathy group, okay? And I think you can already see that with diabetic retinopathy, this response looks like it's smaller in amplitude. But what I'm really interested in is the oscillatory potentials. And so they are filtered over here from these waveforms. And you can immediately see that the ICEV dim, yeah, these oscillatory potentials are hard to measure, right? They're hard to see, even in the control. So that dim flash is too dim. But once we use this test flash 1, we can see the oscillatory potentials. And the test flash 2, they're very, recognizable there. And then, of course, in the bright. Now, we look to see if there was a delay. And there's a little bit of a delay here in the diabetics. that don't have retinopathy. And then in the diabetic retinopathy group, there is a delay. And we saw a similar thing here. But then here in the ICEP Bright, we only saw a delay in the DR group, not in the DM group. So this data seemed to indicate that, yes, similar to our rodents, we also were able to see some sort of delay in the patients. All right, so we're thinking here about a biomarker. So it's like, okay, well, this is interesting, and maybe you could actually use the ERG for some sort of testing, screening, outpatient screening. But if you've done an ERG, you know that ERGs aren't done everywhere. In fact, they're only really done in academic ophthalmology centers. And the reason is because they're hard to record and they're hard to analyze, right? So if you're thinking about something that could be used by anybody to do screening, you're thinking, yeah, that's not going to work. And we have a similar kind of concern. So if you do, clinical ERG, you dark adapt usually for 20 to 30 minutes. You apply dilating drops. You have some sort of corneal electrode that you're putting on the eye. Here's a couple examples. You have a Gonsfeld, dome that you're presenting the flash of light in. This has a pretty big footprint that you're going to have to put your head into. And then you need an expert to actually analyze this waveform, right? So all of this together just means like, this doesn't seem like a very suitable solution. However, there is a product on the market right now that makes some of these things more accessible. And this is the LKC Retival. It's a handheld device, and you can hold it up to the eye. You can see it has a little window here. And it's often used with a sticker electrode that's just placed underneath the eye. And so this kind of solves a couple things. We wouldn't have to put an electrode on the eye, right? You can use a sticker. The way this instrument is designed is it has a pupil tracker, and the pupil tracker can adjust the brightness of the flash depending on the size of the pupil. So now you don't have to dilate the eye anymore. Okay, so that's advantageous, right? And then we tested, well, how long would you actually have to dark adapt in order to get this dim flash oscillatory potential we're interested in? And it turns out we'd only have to dark adapt for 10 minutes. Well, 10 minutes isn't that long, right? So that all seems a lot more feasible. So Kara Motz in the lab did a clinical study where we recruited some diabetic veterans from our VA that did not have retinopathy. And we're comparing them to non-diabetic controls here. And I'm just plotting the oscillatory potential implicit time. And so you can see the values for the non-diabetic controls. And then here are the values for the patients that had diabetes without retinopathy. And you can see that their implicit times were significantly longer, slower than the controls. Now, what struck me is that when we did the screening, there was 52% of the patients that we screened had delays. So that's pretty significant, right? So it's not like only 10%, 52% actually had delays. So there's a lot of people who are walking around who have diabetes who haven't yet had a diagnosis of some sort of vascular problem that have some sort of functional issue with their retinas. Okay, so this seems good, right? So maybe we do actually have a biomarker that could be used, a functional biomarker. And when you have that functional biomarker, you now have an opportunity to treat at a different time. And it's important to know what are the current treatments that we're using for diabetic retinopathy. And they're all focused on the vasculature, which makes sense. That's how we're diagnosing it. So the current treatments are an anti-VEGF injection therapy, laser surgery, so they actually burn off like the abnormal vessel growth, or corticosteroids. And those are the three major treatments that are out there. And I would really argue that this is mid to late stage disease, right? We're now identifying disease that's happening much earlier, but we're waiting to mid or late stage to actually even do anything. So the goal here is to treat diabetic retinopathy early and hopefully prevent any kind of vision loss. So what we've been focused on is dopamine. So we have discovered, and others have been pointing this out as well, that in the diabetic retinopathy, it seems that there are changes in dopamine. And there was a study that reported that in about 6 months of diabetes in a rodent, you actually lose dopaminergic amacrine cells. And so Harry Yong in the lab, he was like, that's interesting. I wonder if dopamine is it all dysfunctional, or the kind of the metabolism of dopamine at early stages. And so this is a 5-week-old or 5-week time point in STZ mice. And you can see that the retinal dopamine levels are significantly decreased. And so based on this, we decided that maybe it would be useful to give the animals L-DOPA. So you can see here that the metabolic pathway in order to synthesize dopamine. We have tyrosine, it gets converted to DOPA, then gets converted to dopamine. And so if we put in L-DOPA or levodopa, you can see that we can increase this step and get more dopamine. And so this is what Harry Ong, as well as Dr. Moon Kim Han did in the lab. So here I'm showing you data from mice. This is at 5 weeks post-STZ. So these animals had the STZ, and then once the hyperglycemia was confirmed, we started on the L-DOPA treatment. So the black line at the top, those are the controls. Then you can see the diabetic animals that had vehicle. You can see that they have that delay that we've seen in other groups. And then we treated them with L-DOPA. And you can see that here, this oscillatory potential is completely normal compared to the control, right? So there's no delay. So it actually was able to prevent that delay from happening. Now, we also looked at the benefit of this L-DOPA for visual function. And so here are diabetic mice again. Here's our spatial frequency thresholds. The black lines are our control groups with vehicle or L-DOPA. Then you can see the wild type diabetic animals plus the vehicle. That's the red line here. So they have a deficit, as we've seen before. But with L-DOPA, this deficit is decreased, right? So there's some preservation of the response. And you can see in contrast sensitivity, it also had preservation. In fact, even more preservation, but then eventually you do see a decrease. So L-DOPA definitely seems to be having some benefit. But this was all treating exactly when we have, hyperglycemia induced. And clinically, you're not going to be able to do that, right? You're not going to have somebody come in and you're going to go, oh yeah, you're diabetic and I'm going to give you L-DOPA right today. That probably is very unlikely to happen. And people have diabetes for quite some time before they even are diagnosed. So here in these experiments that Kyle Chesler did, we were actually trying to have an experimental paradigm that was closer to what you would find in the clinic, where we made the animals diabetic and we waited until they had an oscillatory potential delay. So you can kind of see that here. So this is with a dim flash stimulus, and you can see that these diabetic animals in blue have delays. And then in the green shaded area, that's when we started the L-DOPA treatment. And so you can see here that the vehicle treated in the dotted line, they continue to get more and more delayed in their oscillatory potential dim flash. But these animals that held L-DOPA, their responses don't. In fact, they get closer to the control in black. So our example waveforms are up here. Now this, I'm showing you here, this dim flash. This is an intermediate brightness flash. And then here is the bright flash. So in this intermediate, we do see, somewhat of a delay here in the animals that are treated with vehicle. And then that is preserved when they're treated with L-DOPA. You can see that in this plot. And then in the bright, you really don't see any benefit, but they don't have any delay either. Well, they have a little bit of a delay, but they don't have benefit from the L-DOPA. So, and then this is the spatial frequency plot. And so here is our vehicle treated diabetics. You can see that they have this, decrease throughout this period. But when we give them L-DOPA, even, at 3 weeks, so even after the diabetes has already started, this kind of prevents, stops, halts that progression, but then eventually it still is going to progress. So definitely some benefit. So we decided, well, let's see what happens in people, right? Let's see, L-DOPA or levodopa is a drug that's FDA approved for patients, right? For Parkinson's disease. So it was very easy to be able to put this in place. So again, Karamatz is the one doing all this work. And she recruited non-diabetic controls and then diabetic patients without retinopathy. And they were randomized to either a low dose Sinemet, which is a combination of carbidopa and levodopa, or a higher dose Sinemet, which is, twice the concentration. The people would take this medication twice a day. And we're doing all the testing using that Retival, that handheld device with a dim flash. So the first data I'm going to show you here is just 2 days after they began their medication. Okay, so they've only taken it for 2 days. And already you can see these oscillatory potential timing has improved. And in the dim flash, really remarkable, also remarkable in the high, although, there's some exceptions. There's a few people that didn't seem to really, didn't do much. So now let me show you the data here from at the 2-week time point. So again, baseline, we definitely see that there's a delay in the oscillatory potentials. At 2 weeks, you can see that the responses are restored to the control levels. Now, that was pretty remarkable. So I didn't expect L-DOPA to have that much of a benefit. I was hoping that we'd see that it'd have something that was, benefiting the patients, but not restoring it back to the control levels. So then what was even more remarkable is we stopped the drug at 2 weeks, and we then had the participants come back at 4 weeks, so a 2-week washout period. And it turns out that they still had significant benefit. So that means that this L-DOPA can't be working in an acute type fashion, right? The levodopa is, not present in the retina probably more than an hour or 2 at the most. So whatever change is happening has to be affecting the cells, immune response, a genetic response. I don't know, but it has to be affecting something. Now, I do want to point out to you that we didn't see benefit in other aspects of this study. So here I'm showing you the bright flash, right? In the bright flash, we didn't see OP delays. We also didn't see any real change from giving the L-DOPA to these individuals. It didn't change that response. And then I also want to show you the fact that we did measure visual acuity and contrast sensitivity in these patients. So really, again, if you're thinking about a screening technique, it'd be far easier to measure visual acuity or contrast sensitivity in patients than putting a sticker electrode on them and trying to do ERGs. And so we wanted to see if we could do this. And the way we did this test is that we actually used drifting gratings using a Gabor patch and 1 of 4 orientations, and we measured contrast sensitivity or visual acuity. Now, we did the drifting gratings because we were interested in doing this more like what the OMR was, right? That, the moving grating, we thought that might be something that you really need here for that sensitivity. But you can see that we really didn't see any significant changes, any significant deficit in the diabetic participants in visual acuity. Here's our contrast sensitivity curve, no difference. And then if we take the peak of that contrast sensitivity curve and look at the contrast sensitivity thresholds across time, we also didn't see any changes. So at least in our hands, we couldn't detect anything in these individuals. The oscillatory potential dim flash response seemed to be the one that was the most sensitive to these changes. Now, I want to remind you of something, which is in our diabetic rodents, when we looked at this dim flash OP delays, we could see progression of this delay, right? So here's our diabetic rats. I've already showed you this plot, and you can see how this gets more and more delayed with the progression of the diabetes. And here's some mice, and it just kind of also shows that, okay, we have this progression to 4 weeks, and then it keeps getting, longer. So what's happening in the patients? Well, we actually have had recently the opportunity to look at this. So those patients that we published from that MOT study, we had them come back. So now it's been like 4.5 to 6 years since they have actually been tested. And we wanted to see what these oscillatory potentials actually look like with them. So remember, they were either dosed with low dose or high dose levodopa for 2 weeks, only for 2 weeks. And then we haven't seen them until now. So here I'm plotting the baseline values. I'm plotting that improvement that they experienced at the 2-week time point with the levodopa. And this is the latest data from them. Now, I was totally expecting to see that this would either be very similar, fairly similar to what they were at baseline, or really I was expecting it to be more delayed based on our rodent data, and they weren't. And that was surprising to me. So, you know, why is this happening? Is it that having just 2 weeks of levodopa several years ago actually provided some reset to this disease process? Maybe, possible. Maybe also that in humans, the progression of these oscillatory potential delays just are not very fast, not nearly as fast as what we see in the rodents. And it maybe takes a long time to see the oscillatory potential delays progress. So the data I have to show you today doesn't have any controls, any non-diabetics or patients that have diabetes who didn't have delays to see, or they weren't on drug to see what happens. So we're trying to collect that data right now from the other participants that we had screened. So hopefully I'll be able to see some different things and show you some different things. We'll actually have this at ARVO this year. So I think a big question here is, how is L-DOPA or levodopa actually providing this protection? And one of the things that we want to be able to know is, well, if you give L-DOPA, are you actually affecting the level of dopamine and DOPAC in the eye? And so this is data from our rodents. We injected L-DOPA, and then we looked 15 minutes after. We sacrificed the animals and processed the retinas through HPLC. And so here you can see just the L-DOPA levels is certainly greater, both in the control group and the diabetic group. And then here you see the dopamine. So the dopamine decreased in our diabetic vehicle. We give the L-DOPA, it increases as you would expect. And in the DOPAC, you can see that this is a metabolite of dopamine, and it also is increased in the diabetics with DOPA. So it's definitely getting there. Okay, so that's an important, you know, check that we needed to know that. We also wondered, well, you know, how much of a deficit do we have here with the diabetes as far as the functional assessment? So in this experiment, Harry actually looked at animals that had a retinal tyrosine hydroxylase knockout. Okay, so they don't have dopamine, they can't make dopamine in the retina. And I'm showing you here spatial frequency and contrast. So this black line is the control animals. And the red line is our typical diabetic animals that had STZ. And then this purple line are these RTH knockout mice. So you can see that just not having retinal dopamine definitely decreased spatial frequency and contrast. And when we have this diabetic animal, their loss of function ends up meeting about what that mutant animal has, right? Okay, so what if we give L-DOPA to those transgenic animals? So that's what I'm showing you here in the green line. So you can see the animals that were in vehicle in the purple, and then if we give these RTH knockout mice L-DOPA, we can improve their visual acuity and their contrast levels. Okay, so L-DOPA definitely can have a direct effect on the retina and improve function. Now, we also looked at what the different dopamine receptor gene expression was doing. So focusing on the D1 and the D4 receptors. And we also looked at tyrosine hydroxylase. And you can see here, I'm showing you data from the controls, the diabetic animals that are either on vehicle or L-DOPA, and then these transgenic animals on vehicle and L-DOPA. So in general, you don't really see much, right? So it means that the diabetes doesn't really seem to be changing the gene expression, but also notice that L-DOPA doesn't seem to be changing the gene expression either. And the only thing that really seemed to change was this one group here for the D4 receptor seems to be significantly decreased. And this is wild type, this is the diabetic animals plus the L-DOPA. And you could imagine maybe that would happen. You have more dopamine just homeostasis, you might be dropping those receptor gene expression because you have more dopamine that's accessible. Okay, but nothing here that's really going to explain how is L-DOPA actually working. Now, we did do another set of experiments that's kind of interesting, where we took and we looked at dopamine receptor agonists, so either a D1 or a D4 agonist. And we looked at spatial frequency and contrast in the diabetic animals. And you can see that in the diabetic animals, when we give them the D1 receptor agonists, they actually have an increase in spatial frequency. And when we give them the D4 receptor agonists, they have an increase in contrast sensitivity. So this is kind of a cool specificity between the dopamine receptors. If you haven't seen this data yet, this is not like we weren't the first to report this. This is in a paper by Jackson et al. That showed that the D1 and the D4 receptors are specific to either visual acuity or contrast. But it worked in the diabetic animals. We have since tried to pursue these dopamine receptor agonists further and see if they could be protective. And we're not really finding that they seem to be as, have as much efficacy as just giving L-DOPA. Okay, so it may be surprising to some of you that L-DOPA had this protective effect, that it could actually work as a neuroprotective agent. But interestingly, L-DOPA has recently been reported to show protective effects in age-related macular degeneration. So there was a retrospective study that showed reduced age of onset of AMD when taking L-DOPA. And here's this data. So you can see an L-DOPA group, an AMD group, and then when they were combined, and you can see the age at which they were diagnosed with AMD is greatly increased. So, okay, that looks interesting. Then a second study also looked at taking levodopa for 4 weeks, and it seemed to improve, and these are in patients with AMD, and it seemed to improve visual acuity. So those are the L-DOPA group is the red bars here. The sham is the first bar in each group. And this is at 1 and 4 weeks. So you can see that they got some benefit in their visual acuity. And then they also found that the L-DOPA seemed to decrease the retinal fluid that was in the retina from these patients with AMD. So again, another evidence of some neuroprotective type function. Now, there's also some evidence that L-DOPA could, or dopamine in general, could have some effect on vasculature. So here's a study looking at D2 receptor agonists in zebrafish, and they found that it inhibited VEGF neovascularization. So a direct effect on the vasculature. And then we also have this retrospective study that showed that levodopa decreased the risk of developing neovascular AMD by 15 to 23%. So, and then finally, I need to tell you that there is a publication that suggests that this mechanism L-DOPA, at least for AMD, is actually because it binds directly to GPR 143 in the RPE. So, perhaps that's happening in diabetic retinopathy as well. I don't have any evidence for that. Or maybe there's some other type of a mechanism. So, there's a growing body of literature that actually suggests that there's neuronal changes that are happening in the diabetic retina prior to these vascular changes. There's, the deficits in the ERG. There's reports of changes in night vision, color vision, contrast sensitivity. There's reports of actual lots of retinal neurons, several different types, photoreceptors, dopaminergic amocrine cells, retinal ganglion cells. and also decreases in synaptic proteins and neurotransmitters. So many different changes that are happening. It's not just about the vasculature. But we can't ignore the fact that we have neurovascular coupling, right? So these two structures are really tied together. So just because we have neuronal changes, it doesn't mean that the vascular component isn't still providing feedback or actually causing these issues. So, and in our lab, we've been looking at functional hyperemia, which is a functional response of the vessels, the blood vessels, to flickering light. And so we can actually measure those blood vessel diameters and see if they change and see how the vessels are responding, how functional they are. And we find that this response in our STZ rats is abnormal at 2 weeks. So in a very early time point. So it's possible that you do have vascular function, particularly in this inner retina, right? That's actually feeding these inner retinal cells, including the amacrine cells that are generating our oscillatory potentials. And that is what is, starting off this process, and making this amacrine cell response abnormal. So to try to get at that in our patient studies right now, we are measuring ERG, but we're also measuring OCT and OCTA and really trying to look at microvascular type of responses. And so the first data we have in this analysis is looking at this foveal avascular zone in eyes that have either OP delays or no OP delays. Everybody is diabetic. And so you can see the superior, the intermediate, and the deep plexus layers here. And we're looking at the circularity of the response, or of this area, the FAZ area, the area of the FAZ and the perimeter. And you can see in the area, we actually do see a decrease in area in the individuals who have OP delays. And we also see that in the perimeter. Notice that that's only in the intermediate capillary plexus. So that's kind of intriguing, right? So that in that intermediate layer that's supposed to be feeding those amacrine cells, maybe there is some sort of changes that are happening very early. All right, one other way that we could try to look at this is to try to do some retrospective analysis. So again, I've been telling you that we have these 10 to 15 years before we see these vascular lesions. So what's the effect of dopamine treatments? So how could we figure that out? Well, because I work at the VA, I can take advantage of the VA clinical database. It's called Vinci, and we can try to probe that. So this has been a collaborative effort with folks in my lab, Kyle Chesler, Dr. Rachel Allen, and Kelly Hogan. And they have gone in and asked, okay, when did somebody first get diagnosed with diabetes? When did they first get diagnosed with diabetic retinopathy? And then what kind of dopamine-related drugs are they on? So here, I'm just showing you these veterans that were taking levodopa treatments after the onset of their diabetes. And you can see that these are the diabetic-only group, and this is the ones that have diabetes plus a dopamine treatment. And then here, on the y-axis, I'm showing you the years between when they got their, they were first diagnosed with diabetes and when they were first diagnosed with diabetic retinopathy. And you can see it delayed at about 1.7 years. So it'd be great if it was like 10 years, but it's a small amount, but there is some indication that taking a dopamine drug might have some benefit. So we're continuing to do analysis on this and trying to write this up. So why is it so important that we look at this, kind of changes so early? And when I think about disease state, I kind of think about it in these sorts of stages. So you have changes that are happening all the time and in our bodies and in the eye. And so you can think of this as like untreated disease, and this is like the progression rate. And we have all kinds of adaptive mechanisms in our body that are going to help to try to dampen that and not, make it become a pathology. In this stage of early pathology, we have the ability to actually reverse it. and you could use some sort of treatment and make a difference. But when we get to late stage pathology, you're really not going to reverse it anymore, right? You may be able to maintain function with rehab, but you're not going to get back to the yellow zone, or certainly not back to the green zone. And so if you think about this as far as diabetic retinopathy go, I think right now, how we diagnose and treat diabetic retinopathy is in this orange zone, the late pathology, right? So we're able to maybe maintain function with some of the treatments, but we're not reversing anything. And what my hope is if we could use a functional biomarker, right, this dim flash ERG, that we might be able to detect really early this pathology. And with a treatment, you might be able to get individuals back down into this adaptive zone. Now, I've emphasized our work on levodopa today, but I actually don't think it's the only thing that could be beneficial. I think really any neuroprotective agent that provided benefit could be useful. And so it could be exercise, TUDCA, there are lots of others. So here's some other work that we've done looking at exercise and TUDCA. And really the point here is just like, here's a diabetic animals that have been ran on a treadmill for 5 days a week, just for 30 minutes. You can see they have preserved spatial frequency compared to the other groups. And this is the oscillatory potential delays, which also showed benefit. And then this is TUDCA. TUDCA is a bile acid, a synthetic bile acid that helps to stabilize the mitochondrial membrane. So a very different mode of action. And here you can see the diabetics with vehicle. If we give them TUDCA, we have preservation of spatial frequency. And here you see the OP implicit time. I know it's a little hard to see because it's small, but they also have benefit from the TUDCA and preserving that delay. So I definitely think there's other ways to restore or prevent these changes. So I'd just like to try to wrap up here. And hopefully I've convinced you that these dim flash OP delays are the earliest functional changes that we see in rodents as well as patients with diabetes. And this dim flash OP implicit time seems to be very sensitive to these alterations in the retina. And I think that that's going to be helpful for not only being diagnostic, but also for being able to monitor treatment effects, because we also were able to see that it changed with treatment effects. And so levodopa appears to be protective for this early neuronal change, but there's definitely challenges to this story, right? So the first is just using ERG in general. So I told you all the limitations of the ERG. It has a desktop Ganzfeld. It's not very practical for screening. The analysis is very complicated. And it's a very small signal. So, it's not this huge, 1000 microvolt signal that's really easy to measure. So I think we need to have some sort of new screening device. So we are looking at trying to create some sort of device that would be able to screen for this sort of response. And the VA has given us some money to do that, to create a prototype. And that is what is being licensed. And we have some new funding that we're about a year and a half into now that looks at patients that are taking drug for 6 months, or one arm is even for 24 months. Okay, so this is going to be really beneficial for two reasons. One is that we're going to be able to see how those OPs change for a much longer period of time. We have two trials, and both of them have placebo groups. But we're also going to hopefully have enough time that we're going to maybe see some of those patients actually develop vascular defects, right? So that's really what we'd like to be able to see, because I think a key question here is, okay, you can do early detection and you can do treatment, but do you really change those later vascular, deficits that happen? That is what we are treating in the clinic, right? And if you can't change that, then all this early detection and treatment isn't really making much of a difference. So it's really important to be able to figure that out. I also just wanted to shout out here to the Mary Tyler Moore Vision Initiative. Some of you, maybe not all of you, remember Mary Tyler Moore. She was an actress. She had her own TV show, but I've learned that some people are too young to remember her. But she had diabetes and actually had diabetic retinopathy and was blind at the end of her life. So her husband, Robert Levine, now is leading the Mary Tyler Moore Vision Initiative to try to establish functional endpoints that are really related to these neuronal changes in diabetic retinopathy. And they have a number of partners that they're working with. So you may hear something about them. And finally, I'd like to acknowledge all the people that have worked in this. It's a very collaborative project with multiple people in my labs. The ones in blue are the ones that are still actively in the lab. Our clinical partners, Andy Hendrick has been just amazing as far as working with him for a number of years, as well as our VA endocrinologists who have really worked to understand diabetes and help with recruitment of projects, of participants, including April Ma, and of course, our funding. I would like to do a little shout out. I know you guys are recruiting. We're also recruiting at Emory Ophthalmology. So if you know anybody who's looking to move south, it's a nice place to be and a nice place to live. Happy to talk to anybody about that. And I'd be happy to answer any questions.
We have a few questions, many questions from the room. So let me just start before everybody settle down. It's a lot about dopamine receptor pharmacology. So you have 5 receptors, and using rat, you will not be able as easily as if you would mice dissect which of the dopamine receptor is responsible. And second, the L-DOPA, it may not be the best starting point. Maybe tryptophan, tyrosine would be a better one, because it is known that L-DOPA also increases the homocysteine. And so this all effect could be related to homocysteine rather than dopamine receptor. Can you comment on that, please?
Yeah, so great points, great questions. Like I feel like we're, even though we've been working on this project for a while, there's a lot more to do, right? So we haven't looked at the different dopamine receptors. I agree that using the mice and looking at some knockouts could help us to try to figure out, which receptors might be involved. We haven't done that yet. I think that's a great suggestion. And we haven't looked at, we've looked at some of the dopamine receptor agonists, but we have not looked at like tyrosine hydroxylase or anything else as a potential, treatment option. So that's an interesting idea and certainly something that we could, look at.
So Michelle is there, right, in your institution, Yvonne?
Yes.
So he has all of those mice. The additional problem you're going to encounter is there are a lot of hetero dimers of the dopamine receptor. So this genetic analysis is not going to be quite trivial. Let's go to question from the Zoom, and Juliet.
Hi, yes, thank you. I'm Juliet Moncaster. I've joined from Boston University, so thank you for opening this up. My question would be, thank you for the very interesting, stimulating talk in the retina, but I'm now thinking, do we know anything about dopamine levels in the brain in diabetes?
Yeah, so great question, and thank you for that. So we do know that dopamine levels are decreased all over the body in diabetes. So in the brain, in the heart, in the kidneys. So it's not just the eye. And that is something that, like you could, think about, well, maybe your treatment should be more targeted just for the eye. But I actually think in this case, having a systemic kind of treatment is going to be more helpful because you might actually benefit all of these organs that have decreased dopamine, and that might help prevent some of the other complications. We haven't looked at that specifically, but I think that's an interesting point.
Tim?
Nice talk. The new VA data is really interesting, but can you discuss what's known about mechanisms between oscillatory potential or neural dysfunction in general and the development of vascular pathology? Do you think that the neural dysfunction in diabetes is causal to the pathology, or are they simultaneous but independent?
Yeah, so I don't have a definitive answer for that. I think that certainly we're measuring a neuronal response, but because of neurovascular coupling, it's certainly possible that there's vascular dysfunction that's happening, and that is leading to this neuronal change that I can measure. And we're trying to, look at that right now and try to understand what exactly is happening in neurovascular coupling. Is there a microglia type of response that's involved in that? Trying to dissect that. I think there's many different angles that you can look at to be able to see, the relationship between these. So, I think it's important for us to understand if the neuronal and the vascular changes are coupled, or if they're not, right? Because if they're not coupled, that kind of leads back to, well, maybe we can do all kinds of things to benefit the functional deficit that we're seeing, but it's not actually going to change the fact that we're going to have later stage vascular pathology that's developing. So we need to understand the mechanism of what we're seeing here.
All right, let's move to Sasha and then Cheryl, and then we'll come back to the room. Sasha?
Hello, sorry for my voice today. Oscillatory potentials are part of the B wave. So when you say they are delayed, is this because the whole B wave is delayed, or they are delayed independently of the B wave?
Yeah, good question. They are delayed independent of the B wave. So the B wave timing doesn't change much. Sometimes we can see changes in amplitude in our response, but usually not until later stages, especially in the rodents that we're measuring. So this is a very selective change just in those oscillatory potentials, which would seem to point to the fact that we have some sort of deficit that's just happening in, those amacrine cells that are generating those oscillatory potentials.
Dr. Kraft?
Yes, that was a great talk, Michelle, as always.
Thanks,
I
Cheryl.
think, the dopamine and the patient population and looking at the diabetic retinopathy in the VA population is really a wonderful resource. I was wondering, in that population of individuals that may be already on L-DOPA from the Parkinson's, is there an incidence or any kind of correlation with diabetic retinopathy in those patients?
Yeah, so we do seem to see that they can still get diabetic retinopathy. So in our analysis, we've looked at patients that have Parkinson's disease and they're taking levodopa for Parkinson's. In the diabetic, veteran population, there's actually a lot of people that take dopamine-related drugs for other reasons. A lot of it is, related to psychosis and whatnot. So that means we have these different populations to look at. So we have looked at Parkinson's patients, though, specifically because often their levodopa, medication profile means that they've taken it chronically, right? So they've taken it usually longer, for a longer duration. And so we've looked at them, and again, we're seeing that kind of benefit in that there's a delay in when they get diabetic retinopathy compared to people that aren't on dopamine drugs as long, but it doesn't seem to prevent diabetic retinopathy.
Okay. And then the follow-up is, I think since you actually looked at the tyrosine hydroxylase deficient mice, you kind of looked at that whole pathway because they don't make any tyrosine or dopamine. So I think that might answer the question that I think Sasha or someone actually raised. So I think it is dopamine. The other question is, when people have Parkinson's and the L-DOPA becomes ineffective because they lose those cells to actually produce dopamine, does that also happen in the eye where you don't have those amacrine cells anymore?
Yeah, so this is a good question and raises a couple things. So we don't have any evidence that the retina is losing dopaminergic amacrine cells at the stages that we're looking at. But there is a report that diabetic rodents will eventually lose dopaminergic amacrine cells. And that is more like at 6 months, which is a long time point for a diabetic rodent. So I can't say that in people, if you're progressing and you have late stage diabetic retinopathy, maybe you don't have dopaminergic amacrine cells anymore. I don't know. I don't know that anybody has been able to look at that. But your question raises something else that I often get asked, which is in patients that have Parkinson's that we give levodopa treatment to, they often have side effects from taking levodopa, right? And so, you know, the concern of, well, is this going to be safe? Like, what kind of side effects are you going to have if you're giving, you know, patients that have diabetes this drug for a long time? And so there's a couple answers to that. One is that, yes, we don't seem to be losing dopaminergic neurons in the brain or the retina. And so that helps. And in patients that have Parkinson's, you know, they have lost their dopaminergic neurons in their brain, and the levodopa is given to them, and then they get this upregulation of dopamine receptors, and then they have to have more concentration of drug, and then they get more, and so they have this cycle, right? And then that's when the dyskinesia and other side effects are really prominent, and then they take a drug holiday, and then they put it back on, and it goes again. So So their situation is just the pathology of their disease is different. There's a couple of case reports that have been published. I've been able to find 3 of them where patients were given levodopa for a very long time, thinking they had Parkinson's disease, and then found out they didn't have Parkinson's, and they never had side effects. So that's a positive. It's like, okay, well, maybe then maybe it'll be okay. And I can tell you so far in our clinical trial, following patients who have taken the drug for 6 months, we have not seen any side effects from that. So yeah, so that's a positive. But yeah, all good questions. Thanks so much, Cheryl.
Enjoyed the talk. Thanks.
Yeah, thank you for your thought-provoking talk. So in some of the slides, I saw possibility of something, and that was in the time around the stimulus, there seemed to be a difference in the background noise. And with the amacrine cells being signal conditioners, is it possible there could be more noise in eyes that are on the way to diabetic retinopathy?
Yeah, so that's an interesting question. It definitely is possible, and in the rodents maybe we see a little bit, but it's nothing that I would have pointed out and gone, oh, like this is a you know, really something. Actually, what I see more is in our clinical data. So looking at the patient data, yeah, that's something that, you know, we're kind of struggling with, is that it is a small signal and you can get quite a bit of noise. And so, you know, our approach has been to try to see if we can improve the whole recording, you know, process and try to get better responses. But maybe you're right. Maybe it's just going to be, there's going to be more noise in these individuals who have diabetes. So interesting. Yeah.
Hi, Michelle. I'm Dr. Zodi. I'm asking questions with perspective to glaucoma. And so I had 2 questions. One, the patient with diabetic retinopathy, you mentioned about the hemorrhage and spots. What kind of vision issues they have? Like they get spots in their vision? And then the second question was, when you are measuring these ERGs, the effect that you're seeing, what kind of cells are contributing to that? Is it RGC or you know, amacrine cells or all other cells? So yeah, thank you.
So patients that have diabetic retinopathy have been described as having, yeah, kind of like kind of random spots in their, you know, like scotomas in their visual field. Is that what you're asking? Is what they're, yeah. And I don't know how accurate that is, but that's certainly what I have, you know, seen in the literature and seen described by others.
So those are like spots in their vision?
Yes, but I've also, like, they're not necessarily round. I've seen, at least the way they've been described is more like, you know, misshapen spots, you know, that, so they're not round, they're, you know, can be any shape. But, you know, I don't know, we all are aware that our visual system is remarkable for compensating, right? So if you have a few of these and you have eye movements, you know, you may not notice that you have these, you know, kind of small scotomas until they get, you know, fairly bad. And then you might be noticing, oh, you know what, I can't really see that well. So this is, you know, one of the challenges our visual system is remarkably good at compensating for things. As far as the oscillatory potentials and what they come from, yes, there's evidence that they come from the amacrine cells, but of course there's, lots of amacrine cells, right? In the retina. So what subtype of amacrine cells, we don't know. So, the other parts of the ERG, the A wave and the B wave, we know A wave is definitely from the photoreceptors, the rods and cones, and depending on the flash intensity, we know if it's rods or cones, and the B wave we know is from the on bipolar cells. So the data supporting that and the experiments that have run is like really solid. But unfortunately, there's not a good way to selectively block out all the different amacrine cells and to be able to see which ones are contributing. So, when we get kind of the pharmacology to do that or other means, it'll be really interesting to see what that looks like.
Would the pattern ERG detect the deficits?
So the pattern ERG has detected some deficits, so people have definitely done that. So that's possible. I don't know how the pattern ERG would compare to our dim flash OPs, right? Because we haven't put those head to head, so I'm not clear about that. But other ERG changes, whether it's multifocal ERG or pattern ERG, have reported. I could point you to some literature that would suggest that patients that have diabetes but no diabetic retinopathy have some deficits in those as well. So, yeah.
Machelle, great talk. Really enjoyed it. So my question is related to treatment of diabetic retinopathy. And so the one way that we know how to prevent microvascular complications is with intensive insulin therapy. So I was wondering if you had looked at either in your rodent model, correcting the hyperglycemia and seeing what the impact is on the oscillatory potential, and also in humans, if you have someone who's uncontrolled and they go on intensive insulin therapy, if that, where we know the vascular changes are going to be ameliorated, how much effect that has on the oscillatory potential changes that you're seeing.
Yeah, those are great questions. We have not tried to correct the diabetes or the hyperglycemia in our animals and tried to do that. I can tell you that we haven't seen any major changes, like with L-DOPA, for instance, like it doesn't seem to change the hyperglycemia levels. In our patient population, we did try to look at this, but the caveat is that our numbers aren't vast at this point, right? So the numbers are small enough that we haven't seen any like convincing trends or certainly no significance between, individuals that seem to be more controlled or less controlled. I agree that ideally you take people that aren't controlled and make them controlled and then see what it is. And in theory, in the diabetic population or in the veteran population, you might be able to find people that are controlled, right? So in theory, we could do that experiment, but we haven't gotten there yet. So I think that's an interesting question. So yes.
Thank you, Dr. Pardue. So a real fundamental question on the measurement. You showed that depending on the stimulus intensity, that you can show a break in the photoreceptor class activation. Is it possible to refine the measurement further based on spectral properties and perhaps flash duration to further discern what is coming from the ERG in terms of photoreceptor class?
Yeah, so so I think that as far as the amacrine cells go, is that what you're thinking? Yeah. Maybe, I guess what I'm trying to think through quickly is, do we know enough about those circuits as far as the amacrine, like if you're trying to dissect out the amacrine cell responses, do we know enough about their responses to be able to take a flash that's slightly longer or some sort of a flicker that would actually allow us to dissect that out? I don't know, I'll have to think about that some more and see if that's a possibility. Typically the ERG is ran pretty standard as far as a very short 1 to 2 millisecond flash, and you're really probing either dim rod circuits or you're brighter and it's cone, or you're doing a flicker type response so you can get cones. So we have these sorts of things, but I haven't seen a lot of people play around with duration unless it's like really long. And then if it's longer, you can actually record other responses, including an RPE response. And so, yeah, it's an interesting question. I don't think.
Okay, we'll discuss it later this afternoon further.
Perfect.
Thank you.
Yeah, thank you.
Okay, Yasmin.
Thank you. Thank you for your talk. Congratulations. I'm sure you and your team are really excited about the work that you're doing. So my question is regarding your handheld device. I was wondering if you did any type of comparative studies between your handheld device and essentially the ERG, like the ERG that's done in the facility.
Right.
And if you saw any supportive evidence that says your handheld device is enough to kind of take your patients from the clinical studies, you know, and treat them.
Yeah, so we haven't, in our diabetic patients, we haven't done that head to head yet. We have certainly done just in control patients compared, you know, the type of response we can get from the handheld versus our other desktop Gonsfelds. And they're very comparable. We are currently running a couple of our tests using the different ERG systems for various reasons. So we'll actually be able to answer that question more directly, I think, in a few more months. But at the moment, I can just kind of hand wave to it. Like, I don't think so, but I'll actually have data to support that soon. So, yes.
Thank you so much for the great talk. So I have a question about the VA patient data investigating the effect of levodopa. So I thought there could be like compounding effect from the like glucose management, the pharmacologic treatment, like smoking history between two groups. So how did you remove the compounding effect between two groups?
Yeah.
Did you use some like statistical method to get rid of it?
Yeah, great question. So, that was the first retrospective study that I've done. It was real eye-opening because it's actually really hard to run a good retrospective study. So what we did a number of things. So, for instance, one of the things that we did is if the main piece that we're looking at is whether or not, you know, how long it takes for somebody to be diagnosed with diabetic retinopathy. You have to be really confident about when they were first diagnosed with diabetes and when they were diagnosed with diabetic retinopathy. So for each of those, we wanted to see a treatment where they had no diabetes and then they got diabetes diagnosed twice. And then we said, okay, yeah, you had diabetes on this date. And the same with diabetic retinopathy, that they had been in the clinic, they didn't have diabetic retinopathy, and then they got DR. And then you look through the chart and you see all these other complications and stuff that they have. And so we tried to pay as much attention, age, HbA1cs, body mass index, we take smoking is actually a hard one to pull out of the VA database, but we did try to look at that, and other things. And so what we did then is we took our non-dopamine treatment group, and actually we took the dopamine treatment group and then we matched them one-to-one with people that would be the same. And so it's actually a matched cohort between the dopamine treatment and the non-dopamine treatment groups. Yeah, but it's doing a good retrospective is it's a challenge. So, yes.
Oh, great presentation. Are these oscillatory potential delays something that is perceptible to a person who has these kind of delays in terms of like delayed perception of something for like a split second, or is there also like other effects that can occur, like delayed reflex, like hand-eye coordination or pupillary response? Can you comment on these?
Yeah, I don't think an individual would be able to know that they have an OP delay. Like I think that would be, just, yeah, you wouldn't notice it. And I don't even know exactly how that would be perceived, like if it was really delayed, and especially because it's so selective, right? So you can imagine once it gets delayed enough, you definitely have other components of the retina that are also being affected. But just as I was commenting earlier, like patients usually don't notice that they have any problems seeing until it's really late stage, right? So that's why you're trying to get people to come in and do these screenings, is because they have no idea that they have any kind of even vascular changes. And we see this in lots of retinal disease, right? That patients don't really realize that they have a problem until it's pretty progressed. And we can talk about RP or AMD or glaucoma or, you know, any one of them. But yeah, but no, interesting question. Thank you.
Yes, that was a very nice talk. So I have two questions. Number one, what's the positive predictive value of the OP delay as a biomarker? And number two, so you showed some data on the D1, D4 selective agonist. What's the relative efficacy compared to the levodopa approach? And have you looked at other related, like adrenergic, serotonergic signaling pathways?
Mm-hmm. Yeah, so we've only looked at the D1 and the D4 at this point, so I can't really answer that. But from initial data that we've done in the lab, it seems that the levodopa works more effectively than the receptor agonist. No matter, and we've kind of tried to use D1, D2, and D4 receptor agonists. D2 receptor agonist is also bromocriptine, which is actually an FDA-approved agonist that's used for diabetes, right? So it's kind of like why wouldn't you, like, you would think that would work, but we actually didn't find that to have much of an effect. So I'm not sure what it is about the you know, levodopa that's providing that benefit. Your other question was about the predictive value of having this biomarker. And I think that the benefit is that if you could tell somebody that they have a measurable problem in their retina, it kind of does two things. One thing is it gives us an opportunity to treat, okay? And as I said, I think there's lots of different neuroprotective agents that could potentially be used. And one of the problems we have in testing neuroprotective agents is we don't have good biomarkers at early stages. So if you take and you looked at, you know, across the wide field of neuroprotective agents for really any probably disease, including the retina, things that worked really good in the lab when they move to the clinic don't work in the clinical trials. And the reason why, my number one reason why, is because we don't have good ways to measure things early. We don't have good early biomarkers, right? So if you're trying something in the lab and it's really early, it works great, and then you're moving to the clinic and now you're mid to late stage disease, that neuroprotective agent is likely not going to give you much benefit, right? It's that whole, like, you know, graph I had of the stages, right? So that's one. So you get a neuroprotective. The second is, one of the things that you know, Philip mentioned earlier was insulin. So we know, and there's been lots of studies that have shown that insulin control really does, or blood glucose control with insulin really does benefit diabetic retinopathy, right, for these vascular changes. So we want patients to control their blood glucose levels, right? So if you, and they're kind of like, what's the motivation for that? So if you have somebody and you can say, hey, you have a defect in your retina, that's a motivator, right? It's like, okay, no, I can measure this. And, you know, we all also hear this all the time, but I think it is true that, you know, patients are worried about losing vision. Like, that's a big deal. And so if that's the threat, and your doctor's saying, I can measure this already, that could be a motivation to keep your blood glucose levels in check. So I think that those are the two benefits for that. So.
Michelle, we have so many more questions, but we will finish with 3 more.
Okay, all right, sounds good.
I'll make this one quick. Hi, Dr. Pardue. Thank you so much for your talk. My question is there a difference between insulin-dependent and non-insulin-dependent diabetes and their risk to go on and develop DR? And with that, I know your STZ model is more of a type 1 diabetes. Have you looked in the type 2 model and see if there's any difference in the OP delays?
Yeah, so we have looked at a type 1 and a type 2 model, and we find that the L-DOPA is also beneficial for their type 2 model. So our type 2 models have been high-fat diet plus a low-dose STZ. And then we've also looked at a type 2 model, the Goto Kazazaki rat, who is also a type 2 model. So that it doesn't seem to be selective just for type 1. I don't have any information about the other kind of types of diabetes other than that at this point. So maybe once we get a large enough database of patients, we'll be able to address some of those other questions. So thank you.
Thank you so much for your talk. You kind of sort of answered my question about the vascular defects at the end of your talk. So my question was,
because you had 4 to 6 years of follow-up study in this 2-week L-DOPA treatment study, have you seen any vascular defects in these patients?
Yeah. So that's a good question. So we had, so at this point, like we didn't have that many, right? So that we've tested that I was showing you the data. I think there was like 9 or 10. And 2 of them have now had diabetic retinopathy, but I don't believe any of the others have. But that's something certainly that we'll have in our ARVO presentation. It'll be, you know, much more defined and quantified. But we didn't seem to see many. But you know, yeah, you know, one of the challenges of looking at the vascular disease is the fact that it progresses so slowly, right? That, you know, NIH or the VA needs to give me funding for like 20 years so I can try to follow people, right, for that long period of time. And, you know, our funding doesn't really help us in following people for a really long time, right? So, yeah, so that's the challenge there.
Okay, then final question, go to Jay-Z.
Thanks for the talk. I think I may miss something about this OP delays. Did it link to any molecular level change in the eyes? It's possible because it's a non-invasive examination biomarker. Potentially, could it link to other diabetes complications?
Yeah, a great question. Not that I know of at this point. So we haven't seen any molecular type of a change, but we're getting ready to do some RNA-seq, and maybe that will reveal something that we're not aware of at the moment. So yeah, we'll see.
We had a great conversation. Thank you very much for coming, and thank you all of you. Let's just join and thank our speaker.